in oversalted lakes, even they can actually grow on oversalted fish, for example. So in our lab, they are growing in such flasks, or they can be produced outside of host cell organism. They separate it, and then through centrifugation, this process is quite straightforward and actually very simple. So bacterial rhodopsin proton pumps, they compose over 80% up to 90% of purple membrane, and all membranes are very neatly organized in 2D lattice. So important to note, characteristic absorption is around 570 nanometers, thickness of membrane is around 5 nanometers, and stability is very impressive because actually it made us to believe it's very prospective material for application as energy for energy systems. So it's stable at temperatures over 80 degrees C in water and up to 140 degrees C as a dry. Wide range of pH values from 0 to 12. It loves high salt concentrations, up to 3 molar sodium chloride, for example. It's very stable and photostable, and basically pretty much good material. And before we started doing it, it's mostly been for decades was used. Opened, discovered by German biologist Osterfeld in 1971, and it served as a very good protein for study membrane proteins in general. So here is important person who my former postdoc Shankar Balasubramanian, who actually started this work with me around, I believe, 5 years ago. So our idea was combine titanium dioxide particles with platinum nanoparticles dots and assemble bacterial rhodopsin pump. And upon light illumination, we believe that titanium dioxide surface actually we have holes and electrons on surface, and bacterial rhodopsin, because it's colored molecule, synthesize whole system to visible light, and electrons produced as a through charge separation are used for reduction of protons to hydrogen via holes scavenged by sacrificial substrate, and whole system is a water splitting system. So synthesis of this inorganic part is quite straightforward. It's also photocatalysis. Clusters of titanium dioxide mixed with precursors of platinum nanoparticles exposed to UV light in presence of ethanol as a sacrificial substrate that results in beautiful, and you see rare, like only few particles, 3 to 4 nanometers in size on surface of titanium dioxide. So if you mix just via self-assembly, most interactions in biological systems are going through self-assembly, not covalent bonding. So self-assembly results in a hybrid, which you can see was characterized by Raman and UV visible spectroscopies. And this system under green light illumination, remember I mentioned 560 nanometers, it's optimum for bacterial rhodopsin. produce over 100 micromoles of hydrogen per micromolar concentration of protein per hour, while under white light, when we have more electrons, it produces over 5,000 units of hydrogen. It's remarkably more than better published results for nanobiohybrids. So to confirm that bacteriorhodopsin serves as a plastic photosynthesizer, we ran photochemical measurements and observed 50% increase in the photocurrent density. Then a photoelectrode is modified by bacteriorhodopsin compared to bare titanium dioxide photoelectrode. So photocurrent measurements presented here under green light at 560 nanometers. You can see while there is no photo response at under green light from titanium dioxide or bleached titanium dioxide bleached bacteriorhodopsin, there is a beautiful flat spectrum of photo response of titanium dioxide modified with bacteriorhodopsin. So which actually shows that bacteriorhodopsin serves as a classic photosynthesizer, injecting electrons to the semiconductor. Next, we actually run transient absorption studies. It's revealing, also confirming that decay lifetime for bacteriorhodopsin, you can see, and bacteriorhodopsin titanium dioxide obviously demonstrate there is an electron charge transfer from biological molecule to titanium dioxide nanoparticles. Next, we decided to actually make the system more efficient and more interesting. It's also interesting. First time I visited India 5 years ago, it was a very big conference at IIT Mumbai, and I met one scientist who said, I asked what are you doing, and he said, I'm developing carbon materials, and this carbon material is called graphene. And I asked him why you're doing it, and in briefly, first time I heard from him about this 2D material, and 6 months later, after our meeting, this person was awarded by Nobel Committee, and it was Novoselov. And we decided to actually to play a little bit with this material, and in reality, owing to its wood, has very good physical properties, and also the thought should be a very good material for interfacing, for more neat interface between bio and inorganic material. So in reality, we used reduced graphene oxide. It's more cheap, and synthesis is also very fun. It's also photocatalysis, starting from titanium dioxide nanoparticles and graphene oxide, and actually precursor of platinum. Again, same conditions. Shiny UV light in presence of electron donors. We actually result in simultaneous reduction of precursor to platinum 0, and also you can see, I don't know how well you can see from distance, flakes of RGO. So idea was again to increase performance of photocatalyst, and we got it. We actually observed remarkable improvement in photocatalytic activity, and at the same time we used much less platinum precursor, means much less platinum material, and here you can see this is photoactivity under green light, and here is under white light. And we present here variations of concentration of platinum, and it looks like we every time to actually shape and optimize photocatalyst, we have to just simply run different concentrations, different ratios to tune and find an optimum. And personally, since I was grown up in Soviet Union, I like this suppressor, CCCP, if somebody understands what I mean. Addition of this chemical actually is used to suppress any proton pumping, because we started after we published first paper, we got questions to provide more evidence that proton pump works for two functions, as a dye synthesizing part, and also improve photocatalytic activity using proton pumping or proton harvesting. And this addition of that CCCP obviously actually reduce photocatalytic activity, showing that contribution of proton pumping process, biological process, is very important. As you can see here, you can we actually increase remarkably amount of hydrogen. And at this moment we thought we just dealing with, since we provide carbon material, means we actually increase surface and interaction between biomolecules and semiconductor nanoparticles, and that's what we thought. Again, current potential curves, as usually we do to demonstrate actually to evaluate mechanism of photochemical reaction. And here it's interesting, you can see under green light, main contributor to photoreactivity is bacteriorhodopsin, while under white light, actually both RGO and bacteriorhodopsin, they contribute equally. Even more interesting, we run EPR of whole system and separate components, and we found very interesting actually mechanism. So EPR provides with two key kind of information. First, it's structural information. It's here. So if you can see that red line is a signal of titanium dioxide RGO hybrid. These two peaks actually results of carboxylic or oxygen-centered edges of RGO flakes, some kind of defects. So upon addition of bacteriorhodopsin, you cannot see. You see this is like peak is sharpened, and you cannot see that two peaks. They are actually it's a healing of effect of healing or repairing of defect edges, defect sites of RGO. So next is actually demonstration of how behavior of the system in the dark versus illuminated under white light, whatever is over 440 nanometers, and upon illumination, black is dark, and red is illuminated. So it's obvious that upon illumination, signals of electrons that corresponds to delocalized electrons in reduced graphene oxide decrease. They pretty much disappear, while signals that corresponds to the localized electrons in P25 actually they appear. This is like evidence of electron injection from RGO to titanium dioxide. Actually, before we published this work in 2014 and ACS Nano, most publications about participation of RGO in photocatalytic reactions claimed RGO accept electrons. In this case, that's what we experimented and demonstrate. Actually, it serves as similar to organic photocatalyst. So I'm not showing all data in here. We actually ran later a bunch of time result experiments and demonstrated RGO injects electrons not only to titanium dioxide but also to bacterial redoxin, to photosynthesizer, and then to titanium dioxide. So upon turning off the light, the signals of titanium dioxide lattice trapped electrons were preserved, means this system is very efficient and electrons and holes are very well spatially resolved and this system is stable. So at the end, if you can see it this, through this column, so our system is really efficient. However, I should add, it's tremendously difficult to compare systems because of different groups. They use different experimental conditions, but at least we are trying to show. So we are very successful. We are developing very successful nanobio system. It's also very important that system is very robust, very stable, and it does not require that all these multiple cofactors, which are necessarily that important for keeping photosystem isolated, for photosystem working. And since I actually again mentioned the epoch of Anthropocene and some fantastic technology achievements, I should also mention synthetic biology. It's another super field which is dealing with control of man over production or over a cell or over organism. And synthetic biology is very promising for energy production by two ways. First way is actually then genetic information in organism like bacteria is edited, whole system is rebooted, and then organism produce fuels or maybe some drugs or very useful chemicals. Another approach, you can actually deal with man-made protein synthesis or with man-made protein materials outside of cell, and it's called cells-free expression. So if we use language of engineers, so DNA, actually it's encoded memory. Input is energy supply, building blocks like amino acids, cofactors. Processor is isolated ribosome complex, and all this system actually, and also we need some lipid or lipoprotein rafts pretending to be membrane to express membrane protein of interest in this system. So what we recently done, we actually demonstrated that all completely man-made system of artificial membrane protein complex interfaced with nanoparticles also can serve as a very robust, stable, and very efficient